From the JAMA Network, this is JAMA Oncology Author Interviews. Conversations with authors exploring the latest clinical research, reviews, and opinions featured in JAMA Oncology.
Hello, everyone. Welcome to JAMA Oncology Podcast. I'm Vivek Subbiah, Chief Early Phase Drug Development, Sarah Cannon Research Institute from Nashville, Tennessee, and also the web editor for JAMA Oncology. It is my pleasure and privilege and honor to host Dr. Roni Shouval from Memorial Sloan Kettering Cancer Center for today's podcast. Dr. Shouval, welcome to our JAMA Oncology podcast. Can you please introduce yourself?
Sure. Thank you, Dr. Subbiah. And it's really a pleasure being here, and thank you for the invitation. So my name is Roni. I'm a physician scientist at Memorial Sloan Kettering Cancer Center in New York, where I also serve as an attending on the bone marrow transplant and cellular therapy services. I lead a lab focused on precision cellular therapy, where we try to understand mechanisms and drivers of resistance and toxicity to immunotherapy.
Thank you so much. So recently, the FDA issued a statement outlining reports of T-cell malignancies emerging from post-CAR T-cell therapies. As we know, CAR T-cells are genetically modified chimeric antigen receptor T-cells, which are potent immunotherapy for several refractory hematological malignancies and also in development in multiple solid malignancies. There have been several high-impact publications directly from the FDA and many colleagues from major cancer centers on this emerging issue from post-CAR T-cell therapy. Dr. Roni Shouval and colleagues have published in JAMA Oncology the paper T-cell malignancies following chimeric antigen receptor T-cell therapy, a pharmacovigilance study. So today we are going to talk about that. So Roni, can you please provide a brief overview of the paper and explain what prompted you to conduct this research?
Thank you, Vivek. Certainly. So, as you noted, Chimeric Antigen Receptor T-cell therapy, or CAR T-cell, is a major breakthrough in cancer treatment. And it involved genetically modifying T-cells from a donor that could be the patient themselves, that's typically the case, or from a donor. And once these cells are genetically modified to express a receptor targeting an antigen is present on the tumor, we infuse them back, and these cells mount an immune response or evoke an immune response against the tumor. Now, these have resulted in phenomenal response rates in a variety of hematological malignancies. Mainly, we're talking about B, acute lymphoblastic leukemia, types of non-Hodgkin lymphoma, and myeloma, which eventually prompted the FDA to approve 6 different CAR T-cell products for these indications. Now, despite high success rates, these are not without toxicity. And most of the studies, or early studies, were focused on early toxicities of CAR T-cells, mainly cytokine release syndrome and neurotoxicity. Cytopenias and infections are also an important type of toxicity, which is more in the intermediate range. But now, as the data on CAR T-cell matured, the first approved product was in 2017, we're getting a better sense of their long-term outcomes. Now, in November of 2023, the FDA launched an investigation into the risk of T-cell malignancies followed CAR T-cell therapy. And what prompted this was about a dozen of cases reported to the different adverse event databases and from clinical trials. By early 2024, there were already 22 reported cases of T-cell malignancies from 5 out of the 6 commercially available CAR T-cell products. While, this does not necessarily imply causation, the FDA did issue a black box warning, which is the highest safety-related cautions for medications for all different CAR T-cell therapies. Importantly, the FDA warning is based on individual reports and do not contextualize the risk. That is, it does not indicate if the risk of T-cell malignancies is in fact higher than what you would expect in this population. And this motivated us to perform our analysis. It's also an opportunity for me just to give a shout out to great work by all the different members contributing to this paper, Ryan Stogard, Kai Grzeski, Adam Goldman, and Miguel Perales, who made this possible. So what we did, essentially, is screen the FDA Adverse Event Reporting Systems, or FARS, for second primary malignancies and T-cell malignancies. And we looked at the time window between 2017 and 2023. Out of million reports that are available in this database of adverse events, we found 8,000 approximately that were related to CAR T-cell therapy. And out of these 8,000 cases, 386 were reports of second primary malignancy, and 11 were T-cell malignancies. So second primary malignancies means that these are malignancies that developed after the treatment. Now, interestingly, the majority of these cases were in fact myeloid neoplasms. Next, to determine if these reports were more common than what we expect by chance, we employed two statistical measures. And without going too much into the details, we looked at reporting odds ratio and another metric called information component 25. And these tools helped us to compare the frequency of these reports among patients who received CAR T-cell therapies to the ones that do not. In other words, we studied whether there is a disproportional increase in second primary malignancies and T-cell malignancy reporting following CAR T compared to other treatments. What we found is a significantly higher reporting rate of both these type of malignancies, the secondary primary malignancies and T-cell malignancies in patients treated with CAR T-cell therapy compared to those who were not, indicating essentially there a higher than expected reporting risk for these events.
You spoke about the FAERS database. Can you give the listeners, again, broad listeners of JAMA Oncology, a brief one-liner introduction about the database?
Yeah, so the FAERS database, or FDA Adverse Event Reporting System, is a tool developed and managed by the FDA with the aim to gather information on safety concerns associated with medications and different therapeutic biological products, I would say.
And also, given the rarity of the T-cell malignancies post-CAR T-cell, what challenges did you encounter in identifying and analyzing these cases within this FAERS database?
You're spot right that T-cell malignancies are a rare event. In fact, we identified only 11 events across a huge number of reports from CAR T-cell patients. And this is exactly the scenario where the adverse event reporting system shines, so to say. But nonetheless, the data in this database still requires a lot of scrutiny and some experience and knowledge or domain knowledge in working with this database. And there was a tedious element of data cleaning and processing, which requires removal of duplicate reporting, dealing with missing data and unreasonable values. Furthermore, the specificity of how T-cell malignancies are reported can vary, with some cases potentially being underreported or misclassified under broader or unrelated terms. So we first employed a very permissive filter, I would say, to screen for cases, and then we manually reviewed these cases. Now, another important decision when performing these type of disproportionality analysis is determining the reference group. So you're asking, if I have a reported adverse event, how likely is this reported more frequently than a baseline? And the question is, what's the baseline? And to assess whether reporting of T-cell malignancy post-CAR T-cell therapy was higher than expected, we needed to establish an appropriate reference group for comparison. This involved identifying reports of similar malignancies in patients not exposed to CAR T-cell therapy, which require careful considerations of these patient populations and treatment backgrounds to ensure meaningful comparisons. If you go a bit on the sort of pharmacovigilance literature utilizing the FDA database, you can see that many times the reference group selected in this analysis is the entire FDA database. So you capture all patients across all diseases, all treatments. Here, we decided to take a more restrictive approach, and we used two types of reference group, one comprising of patients with hemato-oncological malignancies and the other patients with lymphoid malignancies. And importantly, our comparison showing a more frequent reporting of T-cell malignancies, the second primary malignancies, was consistent across these two reference groups, suggesting higher reliability of the signal.
So one question we all have as a group in oncology is there any evidence to suggest a causal relationship between CAR T and T-cell malignancies?
That's a great question. And I think part of the reason that this question arises stems from the process of generating CAR T cells. So typically, a retrovirus or a lentivirus coding for the CAR gene is transferred to the T cell that is extracted from the patient or the donor, and it randomly incorporates to the T cell DNA. This raises a possibility of what we call insertional mutagenesis, where the viral vector used to insert the CAR gene integrates into an area near an oncogene or a tumor suppressor gene. And such an integration could potentially lead to the development of malignancies, such as T-cell malignancies. This is a very rare, and mainly we think currently in hypothetical scenarios with the current CAR T cells, but it's still a possibility. So this, I think, always is a concern when you're giving genetically modifying treatments. And with respect to the T-cell malignancies that developed after CAR T-cell therapy, mind you, this is typically given to B-cell malignancies or lymphoid malignancies that do not have any classic T-cell involvement. We have very limited data on the molecular and genomic architecture of these T-cell malignancies after CAR T-cell. We know that in one case developing after ciltacabtagene autoleucel, it's the BCMA-directed CAR T we give in myeloma, the CAR transgene could be found in the 3' UTR of a gene called PBX2. But it still remains unclear whether this CAR integration actually drove the malignant transformation. The fact that we find it does not imply causality. And then in a recent publication by Gallardi and colleagues in Nature Medicine, they documented a case of peripheral T-cell lymphoma developing only 3 months after axicabtagene ciloleucel, which is a CD19-directed CAR T-cell we give typically for large B-cell lymphoma. Now, the number of the identified CAR T or CAR transgene copies in the tumor biopsy were notably low. And in fact, these clones, the T-cell clones of the lymphoma, could be identified even before CAR T-cell infusion. So I think overall, the jury is still out there with respect to causality, and it's important to keep in mind that patients with B-cell lymphomas and lymphoid malignancies and patients receiving immunosuppression have a higher baseline risk for T-cell malignancies. That means that there may be competing causes or drivers for these T-cell malignancies after CAR, or it could also be a multifactorial process.
Thank you so much for that answer. Considering the substantial treatment burden carried by the CAR T patients, including exposure to prior therapies, how do you interpret the high frequency of myeloid neoplasms in this population? Are there any implications for treatment sequencing or timing of CAR T administration?
Yeah, we should be cautious when interpreting these secondary myeloid malignancies after CAR T-cell therapy. To me, it suggests a multifactorial etiology linked to probably the interplay between the therapeutic intervention, the nature of the underlying hematological malignancies, and patient factors. And you could speculate on several factors that would contribute to development of myeloid malignancies and other malignancies after CAR. First, clonal hematopoiesis of indeterminate potential or CHIP. A notable proportion of cancer patients and older patients exhibit CHIP, which is characterized by hematopoietic stem cell clones harboring specific genetic variants or mutations. And this post-CAR T immunosuppressive milieu, compounded by all this intensive conditioning and previous treatments that we give, can hasten the progression of CHIP to actually full-blown myeloid malignancies. Now, this comes over extensive prior treatment. We know that many of the patients received previous autologous hematopoietic cell transplantation and other chemotherapeutic agents that are associated with malignancies. There could be an immunosuppressive effect of the CAR T cells themselves that would augment or increase the risk of developing cancer down the line. And even the inflammation associated with CAR T cell administration manifests as cytokine release syndrome. It could accelerate to some extent processes such as DNA damage and oncogenesis. So I think it's going to be pertinent to elucidate what are the factors actually contributing to the development of these malignancies and develop strategies to identify these risk of progressions or the patients at risk for developing these malignancies early on.
So, in summary, do CAR T-cell therapies still outweigh the risks of treatment, including the secondary malignancies, for the approved indication? What are the next steps in research, and what is the take-home message for the listener?
CAR T-cell therapy provides durable disease control and perhaps cure even for a proportion of patients. There are certainly risks associated with this treatment, and our findings and other also suggest that second primary malignancy could be one of these risks. But still, the benefit could be substantial. And the question is not as critical in patients that have relapse refractory disease, received multiple lines of therapies, and it's clear cut. We don't have good solution. These patients should get a CAR T-cell treatment. I think with CAR T-cells now given at earlier treatment lines or for other conditions, there is now literature to suggest that a CAR T-cell targeting CD19 are also effective in autoimmune disorder. In this population, we have to think more carefully about the risk of long-term toxicity and second primary malignancies, because they could live longer essentially with their disease, and it could be more of a chronic condition. And the data generated now by studies comparing CAR T-cell therapy to alternative strategies in earlier treatment lines could shed light whether the CAR T-cells really increase or maybe even reduce the risk of second malignancies because we give the treatment earlier, and less treatment exposure means less likelihood of developing a second primary malignancy. Also, when interpreting these results, one should think about whether there is a bias in the time for estimating adverse events or second primary malignancies after CAR T. Now, patients receiving CAR T may live longer, and this gives them more time to develop second primary malignancy. So, this should always be at the back of my mind when considering these results. Overall, I believe that moving forward, we need to vigilantly monitor our patients for second malignancies. We need to make sure that we discuss potential risks and benefits among ourselves and with our patients. And we finally need to think about, are there actually mechanistic explanations linking or showing causality between CAR T cells and T cell malignancies?
Thank you so much, Dr. Shouval. It's been a pleasure and honor talking to you. I'm Dr. Vivek Subbiah. I've been speaking with Dr. Roni Shouval on T cell malignancies following Chimeric Antigen Receptor T cell therapy, a pharmacovigilance study. You can find a link to the article in this episode's description. This episode was produced by Daniel Musisi at the JAMA Network. To follow this and other JAMA Network podcasts, please visit us online at jamanetworkaudio.com. Thanks for listening.